Hello, welcome to Biology for You. In this video, we are going to discuss DNA replication. So, in the series of videos, we have been discussing different concepts in molecular biology. So, in this video, we are going to start this DNA replication. So, basically, in this video, we are going to have what is DNA replication and what are the characteristic features of the DNA replication. So, what is DNA replication? Basically, if this is a parental DNA, if it is undergoing for the process called DNA replication, the genetic content of the parental DNA will be duplicated in the form of two daughter DNAs. So, after DNA replication, it produces two daughter DNAs which are same to the parental DNA. So, that is what the DNA replication. Generally, during the cell division, the genetic complement of the cell must be duplicated so that the genetic complement can be given to the daughter cells equally without any difference. So basically, that is why the DNA must be replicated. So here, what are the characteristic features of the DNA replication? As it is the regular process in prokaryotes and eukaryotes, and it is the lead process for any cell division. It should be tightly regulated, and this must be a complex process, and if there is an enzyme requirement. There are some other kinds of characters for the DNA replication. So first one is the directionality of the DNA replication. So, if the DNA is being copied or replicated, the direction of the DNA replication will be 5' to 3'. That means on a template, that means the DNA, if you take this DNA and the DNA, two strands of the DNA is generally separated, and each strand acts as template, and newly added nucleotides. The nucleotides will be added to grow the newly synthesizing DNA strand, the complementary strand, but in the single direction called 5' to 3' direction. So whenever the DNA replication is carried, the direction of the addition of the nucleotides in the growing chain will be in the 5' to 3' direction. So that is what the directionality of the DNA. And what are the other features? DNA replication is basically called as a semi-discontinuous process. So as we already discussed, there is a directionality for the DNA synthesis, that is, the addition of nucleotides will be carried only in the direction of 5' to 3'. But we know that two strands of the DNA will be in opposite orientations, anti-parallel to each other. One will be in the 3' to 5' direction, and the other will be in 5' to 3' direction. So if the DNA replication is being carried, the two strands of the DNA must be replicated at the same time. But the addition of nucleotides must be in opposite direction. For one strand, it must be in 5' to 3'. So the direction of replication is like this. For the other strand, the addition of nucleotides must be like this, in the opposite direction, that is 5' to 3' direction again. How it happens? So because of the other strand which is actually opposite to the directionality of the DNA replication, this other strand, which is called as lagging strand, will form small localized loops. So these small localized loops will give the opposite direction. Because of the loop formation, the DNA template will also be oriented So here this is 5 prime, 3 prime. So that the addition of nucleotide will be carried in the 5 prime to 3 prime direction, which matches exactly with the directionality of the replication of the DNA of the other strand as well as the DNA replicative enzyme. So because of the small loops that are formed, the DNA replication from the lagging strand is discontinuous. So once after the DNA is replicated in this small loop, localized loop, it will go back into the, and again this loop will be dissolved and the newly synthesized small part of DNA will be incorporated in the growing chain, and again the loop will be formed at the forward position, and again that will give the birth of the other such small stretch of DNA, so that the DNA synthesis process is discontinued. It's not continuous process. It will be produced in small stretches of newly synthesized DNA called Okazaki fragments in the lagging strand, but considering leading strand, this process is continuous because it matches with the directionality of the DNA replication. So hence this process is called semi-discontinuous. Because of the lagging strand synthesis it is discontinuous, because of the leading strand it is continuous. So together it makes the semi-discontinuous process of the replication. So what are the other characteristic features of the DNA? So the DNA replication has the directionality, it is semi-discontinuous, and it is semi-conservative. What is semi-conservative mode of replication? So here this is a parental DNA, and if it is going for the replication, 2 daughter DNAs will be formed, and each daughter will get 1 parental strand, and it will be acting as a template to copy down the complementary strand, which is newly synthesizing strand. So for each daughter they will be getting 1 parental strand and 1 newly synthesizing strand. So the parental DNA is partially conserved among the 2 daughters, hence it is called as semi-conservative mode of replication. So that is the other feature of the DNA replication. And the other character is it is a primer dependent. Basically the DNA replication will not be carried without primer. That means they need some support. This replicative enzyme. So if the DNA replication is carried in this place, the DNA replicase, that means the DNA polymerase enzyme must be come and sit over the place where the replication is being carried, and it should start or initiate the process. But it cannot initiate the process by its own, but it needs a support on which it can be sitting and start the addition of the nucleotides. So for the addition of nucleotides it needs some small part of DNA which is called as primer, which is oligonucleotide. So without primer the DNA replication cannot be carried, hence it is called as primer dependent process. The DNA synthesis cannot be carried de novo, but it needs a primer for initiation. And the other feature of the DNA replication is it is an enzyme dependent process. So there are many enzymes that are employed, even in some of enzymes and some proteins must be employed for the DNA replication to be carried. And it is basically a template dependent process. So the DNA replication will be carried using parental DNA as a template. So they will be just a parental DNA which acts as template. So based on the sequence present on the parental DNA, the nucleotides are being copied. So if A is there in the parental template, then the complementary nucleotide that will be going to be added on the same place, and all these nucleotides will be stitched together in the form of newly synthesized DNA. So the parental strand will act as a template for the newly synthesizing strand. So the DNA replication is template dependent. And what is the other feature of the DNA replication? Basically, the DNA replication is having high fidelity, because as we already discussed, actually DNA replication will be carried only when it is needed, and it should be accurate, very much accurate. Otherwise the DNA characters will change, the genetic nature of the organism may change. That means the genes might be changed or inactivated. So it must be, the sequence of nucleotides must be accurate. Whenever they are going to be copied in the form of new DNA, it will always check for its fidelity. So it is tightly regulated and highly accurate procedure. And what are the other features of the DNA replication? So basically these are the primary features of the DNA replication, and it is having basically 3 stages. So for our understanding, for our convenience, the DNA replication can be divided into 3 stages. One is initiation, and second one is elongation, and third one is termination. So what is initiation, what is elongation, and what is termination? So initiation is the process by which the DNA replication will be started, will be initiated. So the DNA generally will be in supercoiled form, and which is actually hydrogen bonded. The 2 strands are come close together by hydrogen bonds. So basically during the process of initiation of replication, these kind of DNA must be opened wherever it is going for the replication. So one open complex will be formed at small localized position called origin. The DNA replication must be initiated. So at that origin of replication, the DNA replication machinery must be attached. So DNA binding proteins will really helps to bind the DNA with them, and they make some topological changes in the DNA, and also they bring replicative enzymes to the site of replication. So this is the process called initiation. So whatever the changes that made in the DNA, so that the replicative enzyme called polymerase will be brought to the site of DNA replication and starts the DNA replication. That means starts addition of nucleotides into the template. And elongation is the process. It is the second step That means once after DNA replication got initiated, it should continue. So addition of nucleotides must be continued and until they reach some termination point, until the whole DNA got replicated. So until the whole DNA got replicated, the addition of nucleotides must be continuous. So that is what the elongation process is. The extension of the DNA synthesis is called elongation. And what is termination? So once after the whole DNA got replicated in the form of two daughter DNAs, then the process must be terminated. At one point, the process called termination site, the DNA replication must be stopped, and that is called the termination of replication. These are the three stages of the DNA replication. So these are the basic character features of the DNA replication that must be understood before we going for the actual process of DNA replication. So it is the DNA replication have directionality, that will be happen only, the addition of nucleotides will be occurs only in 5' to 3' direction. It is a semi discontinuous process because the lagging strand synthesis will be discontinued. A discontinuous process. The lagging strand will be synthesized in the form of small stretches called Okazaki fragments, and it is a semi-conservative replication. That means the parental DNA will be preserved partially among the daughter cells, and it is a primer dependent because the DNA replication cannot be initiated de novo, and it is tightly regulated and enzyme dependent process. There are many enzymes employed in this process. Again, it is a template dependent, it is having high fidelity, and there are three stages in the DNA replication, that are initiation, elongation and termination. So these are the basic characteristic features of the DNA replication. Hope you understand this video, and if you like, share and subscribe to my channel, and press bell icon for the further notifications. Thank you.